Am I audible?
Yes, sir.
Yes, sir.
Okay. Good afternoon. In discuss characterization of those unique variables and so in today's class, we are going to start a new lesson chapter that is actually kind of continuation to where we left off in our epigenetics course. That is eukaryotic gene regulation. And so I would want you to pay attention to details, and I would also want you to review what for you to review what we have learned in the previous course. So, but you have enough introduction. That will be fine. In today's class, I will discuss.
Sir, your voice is low, I guess.
Okay. Is it low for everybody?
Yes, sir.
Okay. I will leave the meeting and come back again, okay? Let's check. How is it now? Can you please tell me? Or is it the same?
It's better now.
It's fine?
Yes, sir.
Okay, good. Okay. In today's class, we are going to discuss about epigenetics, an introduction to epigenetics. And I think we already had good enough introduction when a little bit of introduction in the previous course at least. And before we go into the actual lesson of nuclear and somatic cell genetic reprogramming, we need to understand a lot about the epigenetics. Otherwise, we will not be able to follow this research paper or the article itself. So if we are looking at any scientific literature, it's important to pick up on the words and the terms and the terminology. Then it will become comfortable to go through the course, go through the scientific literature. So we know about the stem cells. We know about pancreatic cells, bones, and neurons. At least we know that they have different functions, and we know that they look different. The important thing is they have the same DNA. There is the genome. Genome is same in all these DNA, all these cells, and yet they have so much of difference in the way they are, their morphology, their form is different, and their function is also different, right? How can that be that if it is the same genome, how can they have different form and different function? That is about the genome gives rise to or results in transcriptome. What do you say transcriptome? It is the total of all the mRNAs expressed at that time in a given cell or cell type in a given instance under some controlled conditions. And the transcriptome will give rise to proteome. Proteome is the total amount of all the proteins with their quantities within a given cell. So if we look at the cell has two things. One is the physical structure, right? That is the form and its reactions. So there are so many biochemical reactions happening in the cell. They are in compartments, and there is a kind of integrity or networking between these compartments to give rise to a function. Eventually, they will give rise to a function. When you say function, it is about how the interactions are, how the cell is responding to the surroundings. The surroundings has different types of cells, or it could have different types of biochemicals coming from afar, and those are the kinds of interactions. Based on that, the cell is reacting to the environment. If we are looking at multicellular organisms, we have several types of these cells, all coordinating and eventually making one individual, right? So if you have the same genome, how can you get so many different types of cells, around 210 cell types? That is to do with what genes are expressed and what genes are repressed, right? Based on that, you have the transcriptome. Transcriptome will result in proteome, okay? So you can call this part which ones are expressed and which ones are not expressed as kind of program, nuclear program. Because, I mean, it's in the nucleus and it's a genetic program, and that decides what cell should look like. That is determining the cell shape, form, and function, reactivity, and all these other properties. What we are trying to do, or why are we studying all this, is to be able to reprogram them. Our objective since the time we started this is as a concept of regenerative medicine. We want to treat people with deficiencies or defective tissues and or lost tissues. And then how can we make that immunocompatible solutions? In that case, we should be able to take a cell and be able to derive the cell of our interest and then give it to the person, right? If we are trying to differentiate one cell type to another, we are actually we are reprogramming. That is why this chapter has nuclear reprogramming. It has a program, nuclear program. We want to reprogram it so that the cell would change or differentiate into a cell of our desire, right? So it is a technical part, which will come in the later part of the class, this lesson. But today, what we'll focus is on the epigenetics. What makes a program and how does the program work? So there will be numerous examples if you go to the scientific literature. Different labs around the world are working on different genes or locus of human genome, and we are trying to learn the general principles. So epigenetics is the study of inherited changes in the phenotype or gene expression caused by mechanisms other than changes in DNA sequence. If there were change in DNA sequence, it would have been mutation, right? Any changes because of which phenotype is or gene expression is changing those are other than that of mutations or changes in DNA sequence, all fall under epigenetics. Epi means above and genetics. You can think of this as the genome, and the genome is represented here as sequence of nucleotides, right? All biological information is either in the form of sequence of nucleotides or sequence of amino acids and proteins, right? They are determining what should happen and so on. So there are several mechanisms that we will see. There are many examples as well. You will find so many examples where there are alteration in the color. It could be in this in some of the cat varieties also. And that is what we will discuss, one of the examples at least. You know about, you may have heard about X chromosome inactivation. It is something, it's also called as, in this particular example with the cat, you call it as Lyonization, where the point is, at any given time, there should only be expression from one X chromosome per a diploid set. Okay, so that is kind of a rule that is there. You cannot have twice the amount of X chromosome expressed. So if it is a male, we have X and Y chromosomes. If it is female, XX chromosomes. In males, sorry, in males there is only one X chromosome, so no issues. Whereas in females, sorry, in females there are two X chromosomes and only one should be expressed. So the one of the X chromosomes is inactivated and it becomes what is known as bar body. So there are two X chromosomes. There is random inactivation, which means in some cells you may have the green one inactivated, green X chromosome as shown in this figure. And in the other one, you may have the red one or purple one inactivated. So as the cells keep on dividing, some cells that come from here will keep producing like this, and the other cells will keep producing other type of colored cells. Now, if you see that what happens eventually in this particular cat variety, there is an allele for coat color or fur color. One of it is for, say, black pigmentation, and the other one is, there is a, I'll put it as fur, and it is mutated. I'll put it as a star, and mutated, so it does not produce no pigment. It does not produce any pigment, right? So the fur color is here X-linked, X chromosome linked. It is present on the X chromosome. Assume that if this is a functional gene and this is the non-functional gene, if inactivation of the green one happens, that is, it forms a bar body, you have one X chromosome active, but it has a mutated fur color gene. So they will all be white in color. Whereas if the other X chromosome, I will call it as X1 and X2 for reference. This is X2. And if X2 is inactivated and X1 is active, that will produce the black color pigment. And therefore, the cells, all the cells that come from here, they would have a black color producing phenotype. So if you look at these cats, some of the regions are white and some of the regions are black, and some of the regions could be brown based depending on the proportion of black and white and how much they are mixed. And again, this is also, there are multiple reasons to it. I will try to send you a link to one of the, I'll try to send you the link to the Coursera course, Epigenetic Modifications or so. I must have already sent. If not, I will do that. I would want you to try to do that course. You will learn a lot more about just epigenetics. For us, it is part of one small part of our this whole course. In that one, it is a whole course is dedicated to epigenetics, and it's very interesting and well described. You can also put it up in your CV if you have finished the course for it. And when you're doing, when you're applying for higher education, if it shows that you did, it's a added value. Okay, I hope you understood X chromosome inactivation. So X chromosome inactivation, the point is that at any time, you call it dosage compensation. At any time, there should be only one X chromosome that is active or expressed. The other one should be silent. In males, there is only one, so no issues. Whereas in females, there are two X chromosomes, so one of it should form the bar body or get silenced or inactivated. In humans too, there are some conditions associated where the same person has one eye brown and one eye very light brown or so, or green, you can think. In some cases, they may have, in the same eye, they may have two different pigments as well. That's called as heterochromia. It is if one person has two alleles, one functional for pigmentation of the iris and the other one without any pigmentation. So there is also a condition called anhidrotic ectodermal dysplasia, where the sebum or the sweat gland producing enzyme, a gene, is present on X chromosome. And some of them have a mutation, right? If in the male there is, in this condition, the pedigree, if you see X chromosome with a functional gene and Y, no problem. We don't have, it's not related. Whereas in females, one is X chromosome with functional gene for the sweat glands, and the other one is not. So this mother is a carrier. And if they give rise to, if these two give rise to children, as long as they inherit the X chromosomes with functional genes for the sweat glands, they would be unaffected, like in this case. If a male gets the X chromosome from the mother, if he gets the defective one, then this whole body will be having no sweat, full of dry, and it has cracks developing, and usually they do not survive until to young youth. But with modern day technology, we can apply these moisturizers and other things, and they can probably survive longer. And of course, you also should remember sweat glands are also important for thermoregulation and also keep the pathogens away and so many other things. The daughter, however, would have received one defective X chromosome if she receives one defective X chromosome from mother and the functional one from the father. Then this, like her mother, would be a carrier. Okay. And what happens is that in the carrier mothers, okay, as I said, because of random X chromosome inactivation, so what happens is some regions of the body are normal, like in this case, where they're moisturized and everything else, whereas there will be many other tissue or these surfaces where they would not be able to produce sweat glands because of the mutation. So they will be of dry patches, which could be treated by moisturizers, and usually they do survive. But if it happens in a male, the whole body is affected, right? Because there is only one copy of X chromosome for a male. So there are some other associated X-linked genes or diseases, and they happen to do with X chromosome inactivation, and there is a possibility of this kind of mosaic type of formation, effect on the body. So now coming to epigenetic mechanisms, there are 4 prominent ones, out of which 2 of them, the DNA methylation and histone modification, we have discussed, and those are the well-characterized and best understood ones. DNA methylation, histone modification, you can just say, which includes acetylation and methylation and also phosphorylation. And the third is chromatin organization, about how a chromatin is organized. Is it tightly packed? Is the DNA or the genetic elements available for, are they accessible to the proteins, other proteins, regulatory proteins or not, and so on. And then there is nuclear architecture. Within the nucleus, the DNA could be arranged in different ways. Towards the periphery, they're usually not expressed. They are usually silenced DNA. Within the core or certain regions of the nucleus, you would have transcriptionally active regions. That is where the genes to be expressed are usually there. That is where they are present. Okay. And I also want to ask you a question. What is nucleolus? What happens there? You know nucleus. I'm asking about nucleolus. What is the function of it? Anybody? If you didn't know, it's worth trying to find out. Every parts of the cell and their functions we should know. Okay? Try to please find it out. So the first part is, today what we will discuss is DNA methylation and histone modifications. So DNA methylation happens in something called as, within regions called as CPG islands. C, cytosine, guanine, and P is the phosphodiester link between these two, C and G. So there are several of these CG, 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 and so on. Like this, you will have multiple or CGs, so many of them. Usually they are present in the 5' side of the genes, but they could be present anywhere. So if this is an open reading frame, and that is a gene, for example, and this is promoter, this is the 5' side, and here on the 3' side. This is where CPG islands are there. It's not like islands or something. It is, those regions are rich in CG, and those regions are likely hotspots for methylation. And where does methylation occur? It happens on the 5' side. 1, 2, 3, 4, 5, and 6. Methylation on the 5th carbon of cytosine. That is a predominant one that is associated with epigenetics. There are about 50,000 CPG islands in the genome, and many of them are associated with housekeeping genes, cell type specific genes, and so on. And there are also many genes that are unmethylated. So as we will progress with more of X chromosome, inactivated X chromosome and imprinted genes, we will learn a little more there. But right now, at the fundamental level, what we should understand is CPG islands are GC-rich regions that are often present they're present in regulatory regions, maybe mostly 5' or near the promoters of several genes. And they can affect how gene expression happens, if it happens or not. So DNA methylation has within the CPG islands, and the second part is, I would also want you to remember this, right? That is, methylation happens on the 5th carbon of cytosine. So DNA methylation has several consequences. It can repress transcription. It can change the local histone modification patterns. It can change the chromatin architecture as well, as we will see in the next slides. And it has a lot of potential in reprogramming or making a program. That is how it is also involved with differentiation. What I want you to right now remember is that throughout the evolution, if there is a DNA, okay, GA, GC, GC. If I'm saying that there is a protein like the DNA like this with a sequence like this that is given here, C, B. Yes. There are several proteins that can interact. For example, when they interact, there are proteins that interact, have protein interactions could be DNA specific. They could be RNA specific. Okay, they could also be sequence specific. So if a if a protein is DNA binding but not sequence specific, what do you think major interactions are with? Major interactions will be with so this is the phosphodiester bond, right? I'm putting the phosphate, ribose, and other things that are on the deoxyribose on the outside, and towards the inside we have the bases here, right? So when you say DNA specific binding proteins, then most interactions are with the phosphate and the deoxyribose. They may be interacting with the bases, but it's not so important. If you talk about RNA specific RNA binding proteins only, without non-sequence specific, then they are interacting with phosphate and ribose, right? Because that is the difference between ribose and deoxyribose, is one of the differences between RNA and DNA. When you say it is proteins are binding DNA, if you say DNA sequence specific, then it would mean that they are interacting with phosphate, they are interacting with deoxyribose, and they are also interacting with the bases, right? Say the sequence that I've given here, if a protein is binding here, that protein may not bind if that base, if cytosine is methylated here. Say if methylation is done, the protein, these methyl groups may interfere with the interaction of these proteins with this DNA, right? That is how DNA methylation plays a role in interactions of proteins that bind to that particular part of the DNA. Okay, keep that in mind, because some proteins can bind to unmethylated DNA, only to unmethylated sequences. Some can bind only to methylated ones. Some proteins do not care whether the DNA is methylated or not. It all depends on the interactions of the protein with the DNA, and if these methyl groups are required for the stable interaction or not. Okay. So now what we are going to see is a sequence of events that will describe what is given here, how DNA methylation could influence the repression of transcription, and then which may proceed to histone modification patterns, and then chromatin structure as well. Here is a representation of a DNA with the promoter, and it is on because you have the enhancers, and these are the activators that are bound. And the activators will allow the binding of the general transcription factors, RNA polymerase, and therefore there is transcription here. If the activator is removed, then there will not be any transcription. It is usually off. It's not as much or very little. And if that is the case and methylation of the CGP islands here happen, if the DNA gets methylated, then it is there is no way repressor is going to bind here. That will not happen, because repressors are sequence specific binding, but they cannot bind to methylated DNA. So the repression is even more, that is the gene expression is much more suppressed. And then there are certain proteins that recognize and bind to methylated DNA. And because the DNA is methylated in this stage, those proteins coat this DNA these proteins that bind to methylated DNA. These proteins also do something else. They recruit other enzymes, which such as histone deacetylases, which we will discuss in a few slides, and chromatin remodeling complexes. So deacetylation, usually histone acetylation, if you recollect from our previous course, we will remember acetylation is associated with increased expression. So these histone deacetylases remove all these acetyl groups from the histone tails. So transcription is furthermore turned off. And you will see that the gene is completely silenced, or that region is completely silenced. Chromatin complexes may turn the euchromatin. This is euchromatin, right? When we were discussing about karyotyping, I discussed some regions are highly stained and some regions are very lightly stained, and so on. The regions highly stained or densely stained are referred to as heterochromatin. Sorry. Heterochromatin, and this is euchromatin. Euchromatin is expressed, heterochromatin is repressed regions of DNA. 1 second. Sorry. So what happens with these chromatin remodeling complexes is that the DNA that was something like this might become something like this, that is much more densely packed. So that is how DNA methylation could influence the chromatin architecture as well. Give me a minute, I'll get some water. Yes, sorry. Can you hear me? Yes, someone, can you hear me?
Yes, sir.
Sir.
Yes, sir.
Okay, okay. Good. Let's move ahead. So today, this will be the last slide, and it's a little bit complex. In the sense, what I might have given you an impression here that DNA methylation would always result in repression. I don't want you to have that in mind. I want you to look at the scenario differently. As I said in this previous slide, DNA methylation, what does DNA methylation do? It affects the way proteins interact. That means the consequences of DNA methylation can be many different things, and they will depend on where in the locus, where I would just put it as the locus of the CPG. Okay, where is it present? The genetic architecture of the gene, that is in the question that we are looking at, whether that is going to have consequences. Third is the type of protein that is interacting. When I said type of protein, I meant it could be an activator that is interacting with the DNA. It could be a repressor. It could be an insulator. Many different types of proteins are there, right? So let's now take that look here. Most simple one that we have discussed is if there is DNA and this is the gene and there is a promoter, CPG island is within the CPG, within the promoter. And if that region is methylated, there will not be any expression of it. If it is not methylated, there will be expression. Why is that? Because the general transcription factors and many other things, they can interact only to unmethylated DNA. They cannot interact with methylated DNA. And look in B. Now there is a variation to this, right? There is a genetic architecture where there could be one gene here and one gene on the complementary strand. And there are both of the promoters have CPG islands. At any given time, one of it is repressed. Say here there is methylation, therefore transcription will not occur. In the other allele, you may have transcription methylation here, so there is no expression from here, but you have expression from the other gene. In the figure 3, this is a little bit complex in the sense that there are two genes. I will call it as gene A and gene B. And we have an enhancer here. This is an enhancer to which an activator binds, the green one that is shown here. So the enhancer can activate both either gene A or gene B. It is an activator for both the genes, but it can only activate at any given time only one of the genes. And whether to activate A or B is determined by another protein, that is a repressor here in this case, which if the repressor binds here, the activator cannot reach gene A. Therefore, it will only activate, it can then activate gene B, and you'll have expression of gene B, but not the expression of gene A. On the other allele, what may happen, say for example, if methylation of the CPG island happened. Remember that there is no CPG island here in A, but there is a CPG island within the regions of B promoter. So if this CPG island is methylated, there is the repressor cannot bind to methylated region here. So the activator can activate gene A, can attract, bend, and interact with A, and can cause expression of gene A. Gene B is repressed for two reasons. One of it is because there is methylation, if it is interfering with the promoter elements as well. The second reason is that there is no activator interaction here. That is another reason why there will not be expression of it. Okay, so in all these three, what we have discussed is DNA methylation is causing gene repression, but that's not always the case. If you look at gene D, you have to pay attention to where is the CPG island. CPG island is somewhere else, not within the promoter elements directly, promoter regions. In all the other ABC, if you see, the CPG island is associated with the promoter elements. Whereas in this case, it is at a different location. It is in a location where a repressor would bind. And this is a silencer, okay? Sorry, insulator, I forgot. This is the repressor. So if the CPG island is unmethylated and repressor binds, it will interfere with the transcription of this particular gene. But if the CPG island is methylated and the repressor cannot bind here, then what will happen? There is nothing to repress transcription from here, so transcription will happen from this allele. So what we should keep in mind is the CPG island, where is it located, and the genetic architecture. And the third or important part is what type of protein is binding to that to the CPG island at that place. When I say CPG island, it is not a certain strict sequence actually. It is mostly about they are highly rich in CG or GC. It doesn't mean this is the same sequence. It could be some AT somewhere too, and again CG like that. You may have variations, of course, but the important thing is if they are nearby any locations where there are proteins binding, then their consequences will be different. I hope you remember this figure, right? From the Watson book that we have studied in the last semester. There are several histone modifications. I'm not going to take this up. I'll just want to review what you may have learned. Histones, these are called as core histones. I want you to help me out. Can you tell me what are all the histones here? What is the linker histone?
So linker is H1.
That's right.
A core is
Core histone.
H2A, H2B, H3, and H4.
That's right. H2B, H3, and H4. Can you tell the composition of a nucleosome?
Histone octamer, sir.
Yes, it's an octamer. That's right. How many? Tell me more details.
Each two, sir. Each core histones will be 2 in number.
Yes, 2H2A, 2H2B, and 2H3H4 heterodimer. Okay, that way also it is octamer, isn't it? So I'm glad you all remember these things. That's very nice. And there are 3 different types of modifications that can happen. This is different from DNA methylation. These are histone modifications. Right now, that is what we are having. And it could be acetylation, methylation, or phosphorylation. I want you to tell me, what does K stand for? The arrow mark that I put. What do you mean by K23? Can anybody help me out?
Amino acid leucine, sir.
Okay. Anybody else?
Not leucine, lysine.
That's right. L is for leucine. This is lysine. Okay, what is 23?
Position, sir.
Yes, position. And proteins, always the position is counted from amino terminal to carboxylic terminal, or N terminal to C terminal, 1, 2, 3, and so on. So if somebody says H3K23, it means lysine at position 23 of H3 histone tail. What does R stand for?
Arginine, sir.
That's right, arginine. And S is for?
Serine.
That's right. And you see, what are these types of amino acids? What is serine? What is special about it?
OH.
That's right. It's a hydroxyl amino acid. What about arginine and lysine?
NH2 in the terminals.
Yes, they are basic amino acids, right? Why? What is the significance of basic amino acids there? Histones are rich in basic amino acids, at least on their surfaces. Why is that?
So DNA is acidic, so to bind the DNA.
That's perfectly correct. So DNA is acidic. Why is it acidic?
Because of the nucleic acids.
What do you mean by that? Which part of the nucleic acid is acidic? Anybody? Just answer. It's a simple question. Nothing complicated in that. What are all the components of DNA? Can we break it down? We have nitrogenous bases. We
Phosphate
have,
group, sir.
that's right, deoxyribose, and we have phosphate group. The phosphate groups are highly negative, and they are the acidic components there. And the amino acids that we are talking about, arginine and lysine, the histones are rich in them because they have amino group which are positive, which have the positive charge. And that's why their interaction is much more better that way. So I'll stop here, and I want you to have a look at this particular part in your syllabus somewhere in the last course, whatever we discussed. It will be nice if you can have a look at it and come, because we will be talking mostly about that itself. Okay? So try to read about the chromatin architecture and other details a little bit. Okay, any other questions or doubts? If not, we'll take leave now. Okay, take care and have fun. You please discuss on the WhatsApp group about the date for final as for the CA exam, and then we will decide one.